My name is Kevin Edwards, and today I want to take you on a little tour of the Lightpath for our confocal microscope. So this is a Leica SP8 confocal, and that's housed in the core facility for imaging in the School of Biological Sciences at Illinois State University. And here is our website. I'll just briefly point out the website here has my contact info if you're interested in using the instrument. We have the full specs of the scope here, a few images, and then we have a page of videos, and I don't have a lot of videos on there at this point. I'm going to try and develop some over time. But what is there now is a brief overview of what I'm going to say today. So today I'm going to expand a little bit on what's mentioned here in this overview. I will not get into some of the applications, and those are described in these other two videos. So you can check those out for more info. All right, so here's the SP8 system. And what you see here is the microscope, but most of what we're going to talk about today is not happening at the microscope. It's happening next door in this scanner box that's attached to the side port of the microscope. Then we'll also talk about the lasers, which are hidden away and not visible here, and they're feeding in to the scanner box. All right, so we'll do a little dissection of the scanner and talk about the overall light path, just as a summary. I will go into a little more detail about the main laser, the white light laser, and how it's controlled, and then how it's scanned over to the sample. So here are a few points about the light path. And we're only going to touch on these first three today. And then we'll just see in overview form the pinhole, the prism system, and the detector system. All right, so here's a schematic of the scanner. Now, I think that every Leica SP8 that's made is made slightly differently. And so the generic diagram from Leica does not necessarily match all systems. So I did my best to try and Photoshop in and out of their standard schematic the things that are specific to our system and that are the most relevant for the user. All right, so let's start here with the excitation side. So on the SP8, we have a set of 4 lasers. So here we have the white light laser and the argon laser. And these each produce multiple wavelengths. And so for the user to select which wavelength they want to send to their sample, there's a special instrument here called the Acousto-Optical tunable filter. And so this allows you to select exactly which laser line will make it through into the system. Then there are 2 other lasers, and these are at set wavelengths, 440 and 405 nanometers. So these lasers are then coupled into the scan head. They pass through a beam splitter and then down to the scanner. Now the scanner will scan the laser light in a raster pattern across the sample using very rapidly moving mirrors that we'll see in a minute. Then that excitation laser light passes through this rotator that allows you to rotate the field that's being scanned on your sample. Then the light leaves the scanner and heads to the microscope. And there it gets directed through the objective and onto the sample. So let's suppose that your sample is GFP, green fluorescent protein. In that case, you're going to select blue light either from this laser or this laser. Going to set the AOTFs to direct that blue light only into the system and down to your sample. The blue light's going to excite the GFP, which will then emit green light. Now that green light is going to emit in all directions, but only the green light that's able to be collected by the objective will go back into the system, back through the same path, through the rotator, through the scanner, and then enter the beam splitter. Now the beam splitter will be set up such that the blue light gets directed to the sample, but the green emission light will pass right through it and pass into the remaining part of the light path here towards the detectors. All right, so the beam splitter is very important. It allows you to use any laser line that you want directed to the sample, and then allow other wavelengths to pass through and hit the detectors. So after the beam splitter, you go through the pinhole, and this is where the confocal effect happens. So this is where the out of focus light will be blocked, and only the in focus light will pass through. This is what gives you your optical sectioning on the confocal. Then you have an optional filter here that can help to suppress some of the laser light that may be bouncing around in the system. And then you head to the detector array. Now, a special feature of the Leica brand in particular is a prism at this point. So whatever wavelengths are being emitted here are all collected and passed to the detectors. And now that collection of different wavelengths, your GFP, autofluorescence, other dyes, for example, might all have different wavelengths passing into this prism. And then those will be spread out and separated. So that now you can grab each of those different components. You can grab the GFP portion and grab a secondary antibody that's labeled with a different type of dye, for example. And you can grab those out of these different positions of the rainbow. And so that's done by taking the emission light and spreading it out into the full spectrum. Now at this point, that rainbow of emission light gets divided up with a system of mirrored gates. And so different fractions of the rainbow here can be directed to different detectors. We have a set of 5 detectors here. That includes 2 photomultiplier tubes and 3 hybrid detectors, which are very high sensitivity and low noise detectors. And so you can basically select any portion of the rainbow and send it to any one of these detectors. All right, so that's the basic confocal light path. All right, so I'll just mention here another offshoot of the light path, and that is the transmitted light detector. So the laser light that passes through the sample can also be collected, and that will give you the equivalent of a standard bright field image that you might see through the eyepieces. So this is not the fluorescence emissions. This is just the laser light that's passing through the sample and basically showing you what the sample itself looks like. And since we have an inverted microscope here, that light, that excitation light, will be passing upwards through the objective, through the sample, and so the transmitted light detector is up here. So most of the confocal light path is all below the stage, and only the transmitted detector is above the plane of the stage and the plane of the sample. Right, so that gives you an overview of the light path. And so now we'll take a little bit of a closer look at some of the first few elements here. This is just a summary of what I just said about the trip of the excitation light to the sample and the emission light from the sample back to the detectors. All right, so now we're going to talk about the white light laser. This is a really remarkable instrument, so I want to go through a little bit about how it works. And so we normally think of a laser as producing a single wavelength of light. And indeed, the white light laser starts off with a single wavelength, but that's in the infrared. It's at 1060 nanometers. So this is outside the range that we can normally use. But that 1060 light is passed through a fiber, and the fiber has small channels in it. And you can see those indicated here. And then this is a cross section of the actual fiber with these small channels. And there's some magic that happens then as the light is passing through those channels. And the specific details are shown here. I'm not going to try and get into that. You can check it out at the Leica information site. But basically the way I would summarize this is that the light is bouncing off the walls of these little channels. And as it's bouncing off the walls, its spectrum begins to expand from a single wavelength to a wide range of wavelengths, right, through a collection of processes that are happening there as it interacts with the fiber. So as it's then passing through this fiber, the wavelengths go from a single 1060 nanometer line to basically spanning across the whole visible spectrum. So by the time the light exits the fiber, you've got the whole rainbow there. And of course, if you see it all at once, if you see all colors at once, it appears white. And so that's why it's described as the white light laser. Of course, white light is not going to be terribly useful as an excitation source for us. So we're going to have to grab individual parts of the spectrum that it's producing and send those to the sample. And so that's what's shown here. Here I just allowed the white light laser to scan across all of its possible output wavelengths. I just took little pictures with the iPhone at the objective, and so you can see that you can get any color from blue out to red. Right, so the output of this thing is pretty strong, from 470 to 670 nanometers, and then it drops off on either side. So the machine is set to allow you to pick anything from 470 to 670, where all those wavelengths are fairly strong. It's not 100% uniform, but fairly uniform across that range. Now, the way the system is set up, you can select at a given time any 8 individual bands out of this full range here from 470 to 670. And the ranges that you collect are just 1 nanometer wide. So you basically got 200 options there, and you can pick any 8 of those 200 options at any given time. Then you can alternate among different sets of 8. So you really have essentially complete freedom to select any visible wavelength range to send to your sample. Okay, so then we have the 1060 light entering the fiber, and then the full spectrum coming out of the fiber. However, we don't want to send all the full spectrum to our sample. So for example, if we have a dye in our sample that's excited by red light, then we only want to choose the red light from the white light laser output and send then only the red light to the sample. We don't want to send every other wavelength because there may be other dyes in there that we don't want to excite. We don't want to kill our cells by hitting them with UV light, for example. So we only want to select the red. Right, so how can we do that? Right, and so luckily there is a substance that allows us to do this, and that's tellurium dioxide. So there's a tellurium dioxide crystal present, and this crystal is transparent to the white light laser emissions. So you send the whole rainbow to it, and that is able to pass through the crystal. But the optical properties of that crystal can be changed by sending acoustic waves through the crystal. So these are actual physical shock waves rather than the electromagnetic waves that you're seeing here. So these little shock waves can actually, as indicated in the diagram here, can actually change the spacing of the atoms in such a way that they will interact with specific wavelengths of light. You can see more about this at the Leica information site here. Here you get the idea that the spacing of the atoms can pick up certain wavelengths of light while others will just pass through. Now, what's important about this for us is that you can electronically change the wavelength of the acoustic waves that are passing through, and therefore you can basically instantaneously switch between different optical properties, different wavelengths being selected, pulled out of this rainbow, and diffracted over to your sample. And so that rapid ability to change the wavelengths you're sending to your sample is very important, because sometimes we want to send only one wavelength in one instant, and the next instant we want to send a different wavelength. So we can view 2 dyes independently of each other. All right, so to illustrate how fast this can happen, I took a picture at the objective of the laser light as it's being manipulated by the AOTF. So what I've done here is to program the AOTF to first grab the red, and then grab blue light, and then green, and then yellow in succession, and then repeat that. And then I just took the iPhone and I panned it across the objective. And so what you're seeing here is about 1 second worth of the emissions hitting the sample and hitting the objective. And so what you can see here is that it very precisely pulls out only the red, only the blue, green, yellow, etc., and it repeats that pattern so that you can then light up a different dye with each one of these wavelengths, and then repeatedly do that. And you also notice that the switching time between each color here, going from yellow to red, the switching time is very, very small, very short. This was set up in such a way that it would change actually very slowly, much slower than we would normally use it on the confocal. But I had to set it up this slowly in order to be able to capture that image with the iPhone. So we can actually do this much faster. All right, so now I wanted to say a little bit about the rest of the lasers and the overall laser array. And for a long time before our SP8, we had an SP2. And so this may be more reflective of some of the older confocals that are out there. And so on the SP2, we had this collection of laser lines. So again, there's this whole array of different dyes that we might want to excite in our sample. But with fixed lines, we can only hit a subset of any of those dyes. Now, the typical fixed lines that you would see are from the argon laser. That's where you get your 488, which is typically used for GFP and FITC. And the argon produces two other very useful lines at 458 and 514. Those coincidentally work well with the cyan fluorescent protein and yellow fluorescent protein. So those have been sort of the workhorse for confocal microscopy and other types of fluorescent systems. And so we retain those lines, that argon laser, in the new system. So we still have the argon laser in our system. Right now with the old system, as would be pretty typical, we had two other laser lines, one at 561 and one at 633 for far red types of dyes, 561 for rhodamine types of dyes. Right, but we were stuck with those lines, and so we could not excite everything right at its peak of excitation. So now with the new system, here's what we have. We have the argon laser still, because that provides us with a nice strong 488 line that sometimes can be useful, and the 458 line, which is just outside what the white light laser can do. And we've added two violet lasers at 405 to light up DAPI, DNA stain, and then 440, which is pulsed, and that's useful for fluorescence lifetime imaging or FLIM microscopy. And then we have our other pulsed laser here, which is the white light laser. And now this one covers the whole range from 470 to 670, and so we don't have the need for individual lasers out here for the reddish types of dyes. All right, then I'll just close with mentioning the scanner. All right, so once our excitation light has made it through the AOTF and through the other Acousto-Optical filter, which is the AOBS, the beam splitter, then the next step is to hit the scanner. And so the purpose of the scanner is to provide your point scanning that you use for confocal. And so this is just a schematic of what's happening here. That laser light is hitting, passing through these two mirrors, and then those mirrors direct the light scanning across the sample, so that at any given instant, you're only collecting light from one point, and then that yields essentially 1 pixel worth of information. And so that's the raster pattern that you see there. Of course, you have to keep in mind that if you have any given object that you're trying to image, you're reducing it to pixels, you're going to be losing something there. So you want to make sure that you collect enough pixels to fully represent the object that you're trying to scan. So you want to typically pick a pixel size that would be on the order of 50 to 100 nanometers if you're working with a very high resolution lens like a 63x lens. All right, so now in our system we have two scanners, and they have their pluses and minuses. So the standard scanner is what we usually use, and this has a variable speed from 1 hertz, 1 scan per second, up to 1800 hertz, and we usually set that at 600 hertz. This produces the widest field of view, and it can bring up very dim signals because you can scan slowly or you can accumulate the signal, and so you can basically see anything that's fluorescing with that scanner. However, if you're dealing with live cells and there's some process that's happening that's very fast, then this might be too slow. And then so you can bump it up from 1800 and go to the resonance scanner, and this is fixed at 8000 hertz. So this is very fast, and you can even double that speed by collecting information both on the forward and the reverse pass of the scanner. And so in that case, it would be 1600 hertz. So this allows you to collect at nearly video rates. You can collect 28 frames a second at a format of 512 pixels squared. Or if you do a strip scan, a narrow scan of 512 by 32 pixels, you can go up to 145 frames a second. That's what this resonance scanner is rated at. So you can see rather fast things happening as long as they're able to produce enough light, enough fluorescence to capture at that quick speed. So with the resonance scanner, you get a very small dwell time. You're only going to dwell for a very short time over any given point. And that will minimize the toxicity of that excitation light, that laser light, towards the cells. So it reduces phototoxicity, good for keeping cells alive when you're scanning them. However, the downside, the trade-off, is that the signal will be rather low. You may have to accumulate the signal by passing across multiple times. All right, so hopefully that gives you an overview of what's happening on the excitation side of the confocal. And later on, we'll post up something on the emission side, and then also how to use the software as well. All right, so any questions, be sure to email me at the website that I mentioned at the top. And thanks for watching.